I think most people know what biological control is, but I'll do the briefest of overviews of biological control for any people out there who aren't aware of what it is. And most of our invasive species come from, well, all of them, not our non-indigenous native species, would come from regions outside of the country, and they're introduced into our country. So in this case, I've just got an example of Brazil, Argentina, where a lot of our invasive species come from. They arrive in their new location, in this case, South Africa, as a pest, and they are free from natural enemies. And when we talk about natural enemies, in this case, it's largely the monophagous, host-specific insect herbivores and pathogens that keep these populations in check in their natural range. And so as biocontrol scientists, what we do is we go to the country of origin and survey in this area for potential natural enemies. And we then bring them back to our quarantine facilities and make sure that they are 100% host-specific, or as near as host-specific as is required for the program, and that they are damaging and that they are necessary to release. So in a nutshell, that's what biological control is. General principles of biocontrol, as I said, is that alien plants invade due to the lack of natural enemies in the new country, and we can call this the enemy release hypothesis. This is just one of many hypotheses as to why plants become invasive, but it's the one that we as biocontrol researchers rely on. The agents are then, as I said, important from the weed's country of origin to reduce the weed's density. What's really important, and that a lot of people fail to understand, or perhaps we don't promote it enough, is that the agents don't eradicate the weeds, they lower their densities. And this lowering of density has to be below some kind of damage threshold that we as biocontrol researchers state is necessary, or as landowners want. And this is the classic biocontrol graph here, where at the beginning, we've got a population density at the beginning of some time period of the weed, which is this black line. And that population is at an unacceptably high density. And it fluctuates, as most populations do, due to various factors, top-down, bottom-up processes acting on their population numbers. Once a biocontrol agent is released, and this is in classical biological control, the biocontrol agent obviously starts off from a very low density. But given the amount of invasive plant or food available for it, to quickly, or sometimes slowly, depending, builds up its population density to such a point that it then starts impacting the population density of the invasive plant. So the invasive plant declines, the invasive plant declines, the biocontrol agent population declines, until we reach a density below a predefined damage threshold. And it is often this damage threshold that we measure our success against. And so we then have this density of our invasive species still in the environment, but at a manageable or acceptable level, and the biocontrol agents fluctuate with it. But probably most importantly is that native vegetation will recover due to reduced competition from the weed, and that's what this line is over here. And so that is biocontrol in a nutshell, and there's plenty of, there's a lot of literature and great literature if anybody wants to read more on that. And I've just put here an example, a very famous example of a biocontrol agent in South Africa, Trichologaster acacia longifolia, which is a gall-forming wasp that galls acacia longifolia, and to such a point now that we don't consider acacia longifolia a significant invasive species in South Africa anymore. The first deliberately planned project in biological weed control worldwide, surprisingly, comes from South Africa. There were some ad hoc programs earlier, before 1913, but the deliberate introduction of the cochineal called Dactylopius salonicus onto a Puntia monacantha in South Africa is regarded as the first deliberate weed biocontrol project. and it reduced populations of Opuntia monacantha to below this acceptable threshold, and the species was then also introduced into other countries where Opuntia monacantha was a problem. And cactus biocontrol is probably what makes biocontrol famous in South Africa, is these really spectacular successes that we've had over the years. And one of them that probably most people know about is the famous Opuntia ficus indica story, or prickly pear, which is a Mexican food plant that was introduced into South Africa as a fodder plant. And in the early 1920s, more than 1 million hectares of the Karoo area around the Eastern Cape, Northern Cape, Western Cape were invaded. That's what 1 million hectares invaded. And by the 1930s, following the release of 2 biological control agents, the infestation was reduced by 90% to just 100,000 hectares. And this weed is still a problem in some areas. If you drive particularly along from Port Elizabeth to Grahamstown, you'll see a lot of prickly pear. And this is largely due to climate and predation constraints. And so we have to use chemical control as a backup. And here is the classic photo of 1939. This is in the Karoo, I think it's near Crawford Net, but I might be wrong. Here is the dense infestation of Opuntia, this little calf of size. And here we are 40 years later, not a prickly pear plant in sight. So really a truly spectacular success, and an example of a program that makes biocontrol famous. So today, throughout the world, biocontrol is implemented in approximately 90 countries. I did read another update that it's actually about 130 countries. Approximately 175 invasive alien plant species have been targeted throughout the world, with the release of almost 500 species of biological control agents. And if we look at South Africa, we have targeted 66 invasive alien plants with the release of 87 species of biological control agents. So it's a worldwide practice. Largely, the biggest or the most active countries have always been the USA, Australia, New Zealand, and South Africa. And over here is just a picture of my little, my favorite biocontrol agent. And if you haven't heard me waffle on about it, I'm not actually going to waffle on about Megamelus frutillaris, the water hyacinth hopper today, but I had to get a picture of it in some way. And so, given the vast scope of this biological control research and the successes of this research, John Hoffman and Cliff Moran published a paper in 2015 looking at what are the patterns of these international symposia on biological control of weeds in terms of the number of delegates, the demographics, and inferences from the debate on non-target effects, which I'm not actually going to touch on today. And one of the things that this paper said is, in view of its long history and the record of extraordinary successes achieved, it is surprising that meetings to discuss weed biological control started only in 1969. And this first meeting was organized by the Center for Agriculture Bioscience International, now called or termed CABI, and back then it was the European station of the Commonwealth Institute of Biological Control in Delamar, Switzerland. And given that the most active biocontrol countries were the USA and Australia and South Africa, it was surprising that the first meeting on biological control of weeds started in Europe. And what Cliff and John said is that these highly constructive gatherings have been sustained to the present day, still without any formal society organization, without any subscriptions or lists of memberships, with no presidents or committee, without annual meetings, minutes, or AGMs. This grouping of like-minded colleagues has no address, bank account, or financial statements. Thus, apart from the scientific papers presented, the only records of what happened on these occasions are contained in the published proceedings. And these proceedings are now all available online at this on this website at biocontrol.org, all the way from 1969, the first meeting, to 2019, which was the latest, or the meeting before the one that we just attended in Delamar, Switzerland. So the first meeting was in Delamar, Switzerland, and then in 2018, the meeting returned to Switzerland. And I think what Moran and Hoffman really brought out was that there's a small group of scientists doing a lot of really impactful research under very little formal organization. And they analyzed the trends over the years. They looked at the countries, the venues, and the dates of these meetings. And you can see that most of the meetings, well, they started off in Europe. They were quite close together, 1969, 71, 73, with very few delegates, 20, 33, 24. It then went to the USA and jumped up to 84. And in Brisbane, it surpassed the 100 mark. But if you have a look at the gender bias, no women, 1 woman, 7 women. And over time, these meetings spread to different countries throughout the world, Canada, Italy, New Zealand. And in 1996, the first meeting was held in Stellenbosch, and with quite a good turnout of 172 people. And I think that's largely because everybody wanted to go to Kruger Park on the post-symposium tour. I'm joking, because of the excellent organization and science in the country. And the proportion of women also increased to about 17%, but still very low. 1999 was the biggest meeting, and that was in the USA. And that was largely because most of the attendees came from the USA and from Canada, so it was quite easily accessible to the North American delegates. And then again in 2014, the conference returned to South Africa, to Coetzee, where 154 delegates came, and there were about 35% of the attendees were women. And if we have a look at the delegates by country of the first 14 meetings, the majority of delegates were always from the USA. So of the 3 out of 20 were from the USA. But over the years, the USA has always been the highest represented country at these meetings. Canada's also always been quite highly represented. Australia, Europe as well has been very highly represented, and South Africa started off with just 1 in the beginning, none for the next 2 years, and then slowly creeps up as more and more biocontrol scientists started to, or as more people started to do biocontrol science, and as funding became more available. And what's interesting here, though, is that the USA, Canada, Australia, New Zealand, and South Africa are the countries that receive the biocontrol agents more than any of the other countries. So we are the ones who rely on getting biocontrol agents from the donor countries, which are regions such as Europe, Asia, and Central South America. And what's interesting is Europe has always been quite well represented, despite the fact that they don't really have a history of practicing biocontrol in their country, but they do a lot of biocontrol research because their invasive species are problematic in South Africa or elsewhere. Whereas Asia and Central and South America, which donate huge numbers of weeds and invasive species, are usually very underrepresented. And over the years, season topics have included the typical biocontrol topics of pre-release surveys in the invaded range, target and agent selection, native range surveys for potential agents, post-specificity testing, which is one of the most important steps, impact assessments, followed by post-release evaluation. But more recently, a number of new topics, new methods have been incorporated as the science has progressed in the light of evolution, ecology, conservation, and climate change, so that we're now looking at the efficacy, environmental safety, and predictability of biological control. What's the role of chemical ecology, modeling, and the omics field? New techniques in taxonomy, including genetic analyses, is a huge field. Impact of climate change. How will these relationships between weeds and their environment and weeds and their biocontrol agents change as climate change increases? Very important is the motivation to use biocontrol in countries where it has not been used before. So convincing countries that biocontrol is beneficial and safe. More importantly, access and benefit sharing of biodiversity resources. And this is largely due to the Nagoya Protocol that was promulgated in 2014. Community engagement, science communication, education feature a lot more in these ICCW meetings. And restoration. What happens once we've controlled our plant, or we've gone below that damage threshold? Do we just leave the plant, or do we need to do some kind of active restoration? And all of this is with the general aim to advance the science and the success of biological control in the world. And so that brings us to the beautiful location where we've just been in Iguazu in Argentina. And this is just a picture of the Cataratas de Iguazu, which are the Iguazu waterfalls and a World Natural Heritage Site, which we were lucky enough to visit on our Symposium tour. Truly spectacular. And this is the at the final dinner. You can see the waterfalls in the background, and they managed to capture the beautiful rainbow. And what's really great, I think, is this logo of these falls, and I'm using the biocontrol, the population density over time biocontrol graph in their logo. And who attended? Well, the majority were from Argentina, which is unsurprising, seeing as it was held in Argentina. But this was a real success, because South America, Argentina in particular, have not implemented classical biological weed control. And so having 35 delegates from Argentina was really important, as well as 7 from Brazil, 1 from Chile, and I think there were 3 from Colombia, and 3 from Colombia. As usual, the USA made up a huge proportion of the delegates, and South Africa came in third. And this was largely made up of staff and students of the Center for Biological Control, for whom I work for. And we were really lucky to receive excellent funding from Rhodes University, from the research office, and then as well as from our some other funding from Environmental Affairs and the NRS, as well as the IOBC, who sponsored some of our students. And as you can see, 13 Australians, 8 from the UK, and slowly went down. And what was really surprising is that there was only 1 Canadian delegate, and usually in the past, Canada has been particularly well represented. So just to break it down, there were 156 participants from 19 countries, representing 64 institutions. 75 people were first-time attendees. And this time there were 70 female and 86 male, so almost getting quite close to that 50-50. Only 19 of the delegates were full-time students, which was a little disappointing, but I think that also just speaks to funding constraints, and something that we really need to look at, because it's these students who are going to go forth and be the future of biological control. If we then go back to Moran and Hoffman's table or breakdown, we can add on Switzerland. So we had 200 delegates in Switzerland and 156 in Argentina. And as I said, 44.9% were now female delegates. And if we go by country, why is that not moving? There we go. South Africa managed to send 30 delegates to Switzerland in 2018 and 25 to Argentina. Just to let you know, this was an incredibly expensive conference to go to, post-COVID, no direct flights. It took a very long time for us to get there, but it didn't stop us from representing. And 46 delegates from Central and South America, which, as I said, is a really promising sign of the uptake or future uptake of biological control in this continent. And again, just to show you over the time that over the last century, as Moran and Hoffman said, a remarkably small group of people have achieved extraordinarily widespread successes through the practice of weed biocontrol achievement that have been of sustained benefit to agriculture and conservation. As I said, the biggest group conference was in the USA in 1999. South Africa hosted a conference here, which is where we had quite a big, a peak in South African attendees, and then again in 2014 in Kruger Park, and this is the beautiful logo for that conference. But in overall, not a very large group of scientists internationally. This was our South African contingent that went to Iguazu, minus 3 of our colleagues from the ARC who weren't in the photo, plus 2 who are our honorary CBC members. This is Gianmarco Minuti from Belgium, who's a student of mine, and Paola Gervasoni, who's from Argentina and also a student of mine. So they're part of the CBC. And a Zimbabwean, this is Gerald Chikawore, who recently completed his PhD with Grant Martin and is now doing fantastic stuff in Zimbabwe that I'll touch on. Our sessions and the number of papers for this meeting, and I think what's important here is it shows how the community, the biocontrol community, is really embracing the most up-to-date, both ecological, theoretical, fundamental, and applied topics. So we had topics on target agents and selection as usual. Climate change, not that many. We still had taxonomy and biological control, prospects for weed biocontrol in South America, ecology and evolution important, implementation of access and benefit sharing measures, which is also something new, but incredibly important. I'll touch on it in a bit. Articulating consensus between science and management has always been an issue. I know particularly in South Africa, where there's often, we're building the gap between research and implementation. How do we integrate biological control, restoration, and management featured quite highly. The future of biological control, importantly, community engagement and education, and then improving biological control studies. So always looking to how to better our science, to reduce those perceived risks, and to improve the level of control that we get. The South African contribution, as I said, we were 25 of 156 delegates, so that's 16%. 3 of us were on the scientific committee of 21 members. We get 28% of the oral presentations. 21 of the 75 talks were given by South Africans, which is pretty significant given that we made up we were a big contingent, but we made up 28% of the talks. Martin Hill gave one of the 11 keynote addresses. We gave 17 poster presentations. Ian Patterson facilitated 1 of 2 workshops. Megan Reed of the CBC was awarded the best student presentation, and here she is receiving her prize. And the best student poster presentation was by Nicolas Salinas, who is of FUEDE, which is the organization in Argentina that organized this meeting. And he is co-supervised by Martin Hill and me. And his poster was first prize with Anna Foltelhauser. So not to take anything away from Anna. So they were awarded a joint prize. So we featured really well in this meeting. And I'm going to quickly go through some of the talks that South Africans gave, which really showcase the kind of work that we're doing and also how we're advancing the science of biological control. So Kim Canavan presented the South African Biological Control Target Selection System. And there are a couple of these systems out there, and Kim's selection went on target plants that don't have control biocontrol programs initiated against them already. And through a series of different matrices and questionnaires, she and her colleagues came up with these high priority target plants. Number one being Spanish reed, and number two being yellow flag iris. And I think probably the most significant thing of this is that the Departments of Forestry, Fisheries, and the Environment were involved with adopting the system because Kim and her colleagues communicated how they reached these top priority plants, what these plants were, what their importance to the government is. And so in 2022, DFSE issued a tender for 5-year contracts, largely based on this prioritization system, which was quite a feat, I think, for biological control. Also then looking at prioritizing or selecting plants, target was Gerald Pikaware's talk on reinitiating classical weed biological control in Zimbabwe. And he showed us that Zimbabwe has a history of classical weed biocontrol, largely against lantana and aquatic weeds. But this has come to a halt, and there's now a huge need to focus on some of these plants that we're focused on and a whole lot of new plants. And so 19 species have been prioritized from all the bioregions in Zimbabwe. And a real win is, I said I wasn't going to show Megamelus, but here it is. Megamelus scutularis, the water hyacinth hopper, has been approved for release in Zimbabwe through Gerald's engagement with the government authorities and through engagement with us in South Africa. And so shipments of Megamelus have been sent to Zimbabwe, and we're really holding thumbs that will see the successes there that we are seeing here in South Africa. If we then move on to taxonomy and agent selection, a lot of work in the past was on, the classical work was on choosing a target, choosing an agent, looking at morphology and original taxonomy. But what's become more and more clear is that we are dealing with challenging species, some that are hybrids, some that are complexes. We are dealing with challenging control agents. We have got cryptic species that might be control agents that we are considering as control agents. And so one of the topics that Megan Reed looked at was looking at host specificity of a South African weevil and a North American hopper for the management of Nymphaea mexicana. Now, Nymphaea mexicana is this beautiful yellow water lily that has got hundreds of, well, I don't know if it's hundreds, but many, many hybrids due to the horticultural industry. And in South Africa, there are very few populations of pure Nymphaea mexicana. Most of them are hybrids with two or more different Nymphaea groups. And Megan did a lot of genetic work on the Mexican water lily in South Africa and found that we have a couple of groups that will be challenging for classical biological control, because how do we know that an insect that has evolved on Nymphaea mexicana in the US is going to be suitable for a hybrid that is made up of mexicana and Nymphaea odorata, for example? So she did some work both in South Africa and in the US, and she looked at, this is a weevil, Vagus longulus, which is an indigenous South African weevil that feeds on our blue water lily, Nymphaea natale. And she found it in the field occurring on some of the Mexicana hybrids and some of the Mexicana plants. And so she decided to do some no-choice testing in to see if we could perhaps use this weevil as an augmentative control agent. And she found that it was damaging across these different species, and so we could potentially use it. And then she also had a look at a classical biocontrol agent, Megamelus toddii, which comes from the US, which is where Nymphaea mexicana is from. And she brought it into South Africa, into quarantine, and tested it against our native Nymphaea species. But unfortunately, this little Megamelus can complete multiple generations on our hybrids and on Nymphaea charlie, which means it's not suitable for biological control. So this hybrid system is really going to play some havoc in us deciding whether we implement a biocontrol program on Nymphaea mexicana or not. Then also looking at choosing a biocontrol agent, Grant Martin has done a lot of work on Robinia pseudoacacia, which you can, I'm sure a lot of you will recognize it. It's become hugely invasive in the high-lying or high elevated areas of South Africa, particularly in the Southern Free State. It's native to the USA in this green area. It's spread outside of its native range throughout the US and throughout Europe. And we've almost got a natural laboratory here because many of the insects associated with it in the US are found in Europe. And so what Grant is proposing is that we look at this natural laboratory to select agents suitable for South Africa. Then looking at classic biological control is just selecting an agent following native range surveys. And here, Ian Patterson and his students, Caitlin Webb, along with Massimo Cristofaro and Patrick Moran, who are from the USA and Italy, are looking at biological control of crystalline ice plants, which is in the A's oasis. So it's one of these little facies that we get on the West Coast that's become hugely invasive in the USA and Mexico, particularly on their coast. And because we offer good value for money projects, probably because our rand is so weak, we often get approached by other countries to do their native range surveys and then native range host specificity testing because it's a lot cheaper and a lot more efficient than doing it the classic way. And what's important about this crystalline ice plant is that it's an ecosystem engineer and that it takes up salt and completely converts the salts into these hypersaline the soils into these hypersaline soils, which then excludes all other plants. And through surveys, they found this beautiful little stem mining weevil, Lixus carinarostris, that mines inside this plant, and pretty soon it will be imported to the US for the last little bits of host specificity testing before it's approved for release. And continuing with the theme of South Africa during surveys and testing for other countries, but possibly a much harder program is this invasive grass biological control. We typically avoided grass biological control as biocontrol practitioners, but why couldn't there be a suite of host-specific enemies? I think the thought's always been that fire and large herbivores drive plant population dynamics, but Ian Patterson's group with Clark van Steenderen and Guy Sutton and Kim and Lennon, a whole lot of his researchers have dug beneath the surface and found a suite of host-specific wasps. And it's been absolutely amazing to watch them unravel their associations, the genetics of these insects. So 15% of grass species native to South Africa have become naturalized elsewhere. And as I said, very few control programs. And what Clark's been working on, what he worked on for his PhD, was these tiny little tetrameter wasps that are associated with a range of different grasses, and looking at their genetic diversity, their systematics, and looking at their prospects for biological control. They're tiny wasps. They are so similar, and so Clark has had to rely on genetic barcoding tools to be able to distinguish between them. And then following on from that, we could then go to post-specificity testing, and this is work that Guy Sutton's done, leading this work with the researchers, and looking at these wasps and doing post-range testing on South African grasses and then Australian grasses. So in this case, it's Eragrostis, which is found in Australia, in New South Wales and Victoria. And the wasp that they're looking at is unlikely to be host-specific because of the close genetic relatedness of the Australian Eragrostis species. Guinea grass, Megathyrsus maximus, which used to be Panicum maximum, also has a potential suite of biocontrol agents, some of them yet to even be identified. And so this whole grass biological control program is really moving forward and building up our knowledge of phytophagous insects associated with grasses. And the beauty of this is that we can do it in the field, and our colleagues from elsewhere don't have to import these agents into quarantine. Looking more at collaborations, we have a north-south collaboration with the case of Iris pseudochorus, or yellow flag iris, which is indigenous to Europe, and with a really great collaboration with Gianmarco Minuti and Iris Steers in Belgium, with Paola Gervasoni and her supervisors in Argentina, and me and my team in South Africa. We've really been able to look at the ecology of this plant, distribution, and look at potential biological control agents for this plant that Gianmarco surveyed for in Belgium. And so this program is moving forward really nicely as an example of a north-south collaboration. If we're looking at post-release evaluations, Megamelis, the water hyacinth hopper, has been released in the US. It's also been released in South Africa. And Nicolas Salinas is based in Argentina, and he's looked at the genetics because the US imported a consignment of Megamelis from Paraguay, which is very different from the original consignment brought in from Buenos Aires. South Africa got the Buenos Aires Megamelis, whereas Florida released the Paraguay Megamelis. South Africa has got incredible success, whereas Florida has not. And what Nicolas has shown, and this was his award-winning poster, is that we are dealing with different haplotypes based on where these Megamelis populations originated. And this could, again, suggest we're dealing with cryptic species. Breakthroughs that we need to work with is how do we claim success? And so often we end our biocontrol programs at the post-release stage, and we don't look at what happens following the control. So our first step, so we've got before biological control, and this is work that Antonella Petruzella and Sam Matitswe are doing with us, looking at once we control an invasive species, and in this case, this is Salvinia molesta. What happens after biological control? Yeah, you can see a bit of Nymphaea mexicana, and this pool is covered in various duckweeds. Is this a success? And so what we have to do is decide, are we merely replacing one invasive species with another, or should we be actively restoring systems both during and after control? And this is work that Antonella is looking at, to look at the short-term direct and indirect effects of biological control on native plant diversity, to determine if we need to actively restore following control. And lastly, I think our biggest contribution at the moment is our contribution to community engagement and and education. Often the criticism of biocontrol is people are worried that insects are going to replace jobs. And so what we've done in South Africa is created a lot of jobs through mass rearing. And so we're taking biocontrol all the way through this value chain, from finding the agents all the way through to mass rearing. And what we've done is created employment opportunities, particularly for people with disabilities. So not only are we creating these opportunities, but we're also getting more insects into the field. And this is work that our mass rearing biocontrol station is doing under the lead of Samela Mzande-Khoza. And then the next step is to allow communities that we work with to understand what biocontrol is, so to demystify the science. And what we've done is developed easy-to-follow protocols for insect rearing, where landowners take on the responsibility. And so not only do they get to be responsible for clearing their systems, but they also get to understand the process of biological control. And that's the community mass rearing that is led by Kim Weaver. We then have also worked with our colleagues in the Southern Hemisphere, in South America, on education, outreach, public knowledge on biological control that can be used in South America, which is, as I said, is typically the donor of all these biocontrol agents, but not necessarily the receiver. And so a lot of work is being done on understanding what the public understand, and then doing these education and outreach activities. And this is, building this bridge between research and the public so that we can improve biological control. And so to close, I think the outcomes of this meeting is that we're doing good science, we're doing good implementation, but one of the things that we have to be aware of is engaging with access and benefit sharing, and this is through the Nagoya Protocol. Martin Hill gave a plenary on classical weed biological control in the Nagoya Protocol. It's time to put it to the test. So in 2014, the Convention on Biological Diversity put this protocol into effect. And the reason for this is to develop a legal framework that ensures fair and equitable sharing of the benefits that arise from the utilization of their native genetic resources. So it's to prevent those countries going in and taking genetic resources or biodiversity resources and the donor country not getting any kind of benefit from it. And so the Nagoya Protocol it was promulgated to prevent biopiracy or scientific colonialism, to prevent the loss of biological diversity, and in particular genetic diversity, and in particular protect indigenous knowledge systems. But the problem is many participating countries were not provided with any guidance on how to go about this. And so what we've done, what South Africa has done, is gone to all the countries in Africa and engaged with them on what their understanding of access and benefit sharing is, in order to ensure that this biopiracy doesn't occur. And we've been able to do this largely through this group, the ICCW, becoming a working group. So this here says a study group because this is on the IOBC webpage. A study group is formed, and only once a meeting is held can it become a working group. And so this study group was formed in Switzerland in 2018, and in 2023, now in Iguazu, the first meeting was held, and so the classical weed biological control is officially a working group of the IOBC, and that allows this access and benefit sharing to be, I hate the word legitimized, but that's essentially what it's doing. And so closing notes then is that South African biocontrol research is in really good standing. We have a very long history of sufficiently adequate funding from several sources. Sometimes funding comes and funding goes, but generally we've been able to conduct the work that we do because we have good funding. We also benefit from excellent collaboration with universities and research institutions, not only in South Africa, but also internationally. And through that, we can develop our science and the science of biocontrol internationally. Our science embraces the full value of the value chain of biological control, from fundamental research on cryptic species to target selection to advanced molecular techniques, all the way through to land use engagement and implementation, which I think is quite unique. We have very good collaboration with government, as we highlighted with that prioritization system that Kim Canavan has developed, and also through the funding that they give us. And we're obviously very good value for money, which is evidenced by this contracted research that we get from other countries. Our challenges: the taxonomic imperative. We need capacity in taxonomy. We need morphological and genetic taxonomy to come together, and we really have to build this, because as we delve deeper into the intricacies of the genetic makeup of both our plants and our species, we really need to understand the genetics. We also need more researchers. We need more implementers. South African biocontrol research needs sustainable funding. So we have good funding, but as I said, sometimes it's not there, and then everything grinds to a halt while the invasive species continue growing. And I think we really need to improve our understanding of access and benefit sharing and our implementation of this. And with that, I will say thank you to all the financial sponsorship that we got, to Fuedi for the organization of this meeting. That was, as I said, fantastic. To the IOBC, they sponsored a few of our students to go. Rhodes, Centre for Biological Control, and the Department of Forestry, Fisheries and the Environment also provided the finances for us all to attend, what was probably one of my favorite BTWs since I've been a biocontroller. Thanks very much.
Thank you very much. Thank you very much, Julie. That was an amazing summary of the Congress, and just thank you so much to you and your colleagues for flying the South African flag high, not only at these congresses, but for, globally for biological control research. And your passion for the field is really contagious, so I can see why
Thank you.
Your group's growing. So at this stage, I want to open the floor. If you have any questions, you can either put up your hand or pop it in the chat. You can just unmute yourself, and no one yet. Well, I'll start, Julie, just by, I really have a passion for taxonomy and barcoding, and to what degree does your group use that for identification? How do we know the biocontrol agents you're using is the right ones? I guess, yeah.
Yeah, well, that's your passion, and unfortunately, it's something I am passionate about, but have very limited knowledge. Ian Patterson pretty much spearheaded that side of our group, and our students are now excellent taxonomists looking at barcoding. Clark van Stenderen is writing his own code for barcoding work. We can use next-gen sequencing tools. We've got a really nice genetics lab at Rhodes, and we also send our stuff away. I think a lot of it comes to Stellenbosch for the I don't know, that stuff that they do with the PCAs and PCR. Which one is it? But yes, I think we are. We keep our students keep up to date with the most recent sequencing methods, the genetics methods. I mean, I've once wrote a paper on AFLPs, and apparently they're not used anymore. So yeah, everyone's very up to date with it, and it's such a useful tool for what we do.
Great, thank you. We have a question from Ashley, who's asking, what are some sites in South Africa where you do releases routinely, if any?
Sure, Ashley, there are a lot of sites. So together our mass rearing and implementation team, and with some of the National Resource Management Program implementators or the biodiversity officers, We release water weed agents around the country. So on the floating water weeds, cactus agents are released all around the country by Ian Patterson's team. The ARC releases agents on a number of weeds that they work on, so pom-pom weed, balloon vine, yeah. So we do a lot of routine releases and then a lot of follow-up monitoring.
Thank you. We had a hand by, is it Ketani? I can't find it again, but you can just unmute and ask. Okay, Tristan, can you please ask your question?
Hi, Judy. Thanks so much for the presentation. It was really, really nice.
Thanks. Nice to hear from you.
Yeah. So I just wanted to ask briefly about, sorry, I'm just going to move out of this room. There's been noise. Can you hear me clearly?
Yes.
Cool. So just about particularly public concerns around non-target effects. Like you said, I mean, there's hesitancy in South Africa with concerns around job losses, but I think more widely, people are kind of just concerned that we're throwing species into new ecosystems, and we don't really have a clear understanding of how that's going to, you know, what the like non-anticipated effects of that are. And I think like some researchers might say that in doing biocontrol, we're manipulating systems that we just don't understand, like to that degree, and to what degree is, you know, a weed controlled just by natural enemies, what degree is it controlled by the entire ecosystem, and you know, all these kinds of questions. So I just thought, what would you say to people who have those concerns in the public and, you know, in research?
Yeah, I mean, that's the question that we get asked quite frequently. How do we know that what we are working on is not going to jump off and is something else? So I think history has shown us that of all the weed biocontrol programs, there are two, I wouldn't say valid, documented non-target cases, because the non-target effects were anticipated, but the agents were released anyway, and that was against thistle in North America, and then the cactoblastis story, which is the cactus moth that accidentally got into North America. It was never released as a biocontrol agent, and those are the two cases that are always cited as the non-target effects and the negative effects of biocontrol. But in terms of future ecological and evolutionary potential, you know, evolutionary changes, we work with what we've got based on the fundamental research. So you know, we look to all the fundamental work done on evolution between insects and their host plants. Conduct our own fundamental research to go forward, and essentially we move as fast as the times and the technology allows us in as risk averse manner that we can. So no one's going gung ho releasing cane toads anymore because the science advanced. But yes, maybe in 100 years' time, someone will say, "Geez, those biocontrollers sure didn't know what they were doing because look at all these cryptic species." Hopefully that won't happen, and hopefully our understanding of how we do our work reduces any chance of non-target effects.
Cool, thank you.
Thank you, Julie. Our next, and possibly might be able to take one more, but we have Darian.
Good afternoon to everybody. My question is very simple. At the beginning, maybe I'm confused, but I understood that most of the pests come from the Americas. If that's so, I mean, do you have any reference for that? Because it was particularly interesting to me. Thank you.
Sure. Yes, there's a lot of references. There's a lot of review papers that show the number of species by region where they become most problematic. So it's South America is obviously largely tropical and subtropical. So species from South America will become invasive in other tropical and subtropical areas. Australia is very high on the list of donating invasive species, particularly to South Africa. Europe is very high on donating invasive species to to the USA. So, if you've got these biogeographic overlaps or climatic overlaps, then that's where the donors come from. But there are a lot of review papers on where the most species, where the most invasive species come from, and where the most biocontrol agents come from, and the countries then that are most invaded.
Maybe just the clarity will be like they might be originated from there, but we don't know how they came here. Exactly.
Yeah, no, they accidentally introduced, intentionally introduced, hitchhikers. There's lots of different ways. No, they're definitely not in. It's not a, oh my word, South America, you're giving us all your invasive species. A lot of them were brought across because they're just beautiful.
No, just because I'm Colombian, I felt a little bit interested in the discussion. Thank you.
Thank you.
Thank you. I can just add to that, there's been quite a large piece of work by Caitlin Faulkner on the transport of invasive species.
Yes.
Yeah. So if you want to look that up. I think I'll just take our last one, if it's okay. David Similani asked, do you think this conference or Congress managed to inspire South America to initiate weed biological control programs?
Thanks, David. I'm going to ask you that because you were there. But I think, no, it's a really good question. And I think there's obviously the biocontrol researchers in South America have been developing biocontrol agents for use elsewhere. And they've seen the benefits of this. And so they certainly have an appetite to initiate weed biocontrol. We're looking at yellow flag iris. We're looking at, I can't think of the other one now. Well, anyway, there's a whole lot. But having some of their authorities at the meeting, I think, was a really good idea to show them what biocontrol science can do. And I think as a result of the conference, yes, I do think that biocontrol might be on a better have a better footing now for the rest of South America.
Thank you very much for that. I think we'll end on this note. It's already past 2:00. So thank you again, Julie. Thank you for your time, and that was really amazing. Thank you everyone for attending. We had 50 participants at one stage, so that's a great turnout. And yes, check our website for future webinars. For July, we have our annual, our Entomological Society meeting in Stellenbosch, so check our website for more details. So have a lovely afternoon, keep warm, and we'll see you next time.
Thank you. Thanks so much, Charlene. Thanks for organizing.
Bye-bye, everyone.
Thanks, bye.